All right, it's recording. We're going to keep trying until this works. What kind of cell do you think this is? All right, what makes you say that? It has dendrites. Yes, these long, spindly things here are dendrites. Why does a dendritic cell have dendrites? What do you think all these yellow dots are? Close, so they can express more antigens. That will happen when they're in the lymph nodes as antigen-presenting cells. They are antigen-presenting cells. Yes?
Maybe because they pick up information and maybe transfer that information to other immune system.
Right, so they pick up information, and the information they're picking up are microbes or anything foreign that has gotten into the body. So all of these little teeny dots around here are representative of extracellular microbes or other allergens. And what the dendritic cells do, macrophages do something similar, is that they sit there in your tissues, mostly your barrier tissues, so your mucous membranes, your epithelium, and they're sampling all the time. So they're constantly phagocytosing any particles that are in the area. And they're checking to see if those particles are something that are dangerous. Because dendritic cells are antigen-presenting cells. So if they detect something dangerous, they're going to take it and present it to the adaptive immune system to say, this is something you need to pay attention to and try to attack. So I'm going to finish today's lecture, or finish last week's lecture, by going through the cell-mediated immune system. We talked about the humoral immune system. That was the B-cell response of the adaptive immune system. And now we're going to move on to the cellular part, it's mediated by T cells. And then I want to talk about transplant rejection. Because transplant rejection really requires a good understanding of T cells. So it's a good way to apply and understand this part of the immune system. And it's those MHC, major histocompatibility complex, which is how T cells recognize antigen. MHC was discovered because it was the thing that causes tissue rejection in transplantation. So we'll try to get through that and then end the class with a little bit about immunodeficiency. So before we get into the T cells, let's just review B cells. So remember, the adaptive immune system has two branches, humoral, which is mediated by your B cells, and cellular, which is mediated by your T cells, mostly the cytotoxic T cells. So in the humoral immune system, we learned about the B cells. And remember that B cells, where are they born? Bone marrow. Where do they mature? Bone marrow, right. So they differentiate into B cells in the bone marrow, and then they go through a process of gene rearrangement to express a B cell receptor on their surface. That B cell receptor is a combination of different gene segments, making it relatively unique. So that B cell receptor can recognize one of millions of different antigens. And in order to prevent autoimmunity, those B cell receptors are tested in the bone marrow against self-antigens. And if they do recognize a self-antigen, they're eliminated because you don't want that B cell receptor getting out into the body. So that's what we mean by maturation. Those B cells that passed the test can now leave the bone marrow, and they're called naive B cells because they haven't yet been activated. So these naive B cells are circulating in the blood all the time. And if I were to draw your blood and look at your B cells, almost every B cell that I look at will have a different B cell receptor on its surface because of that gene rearrangement process. And hopefully none of those B cells will recognize any of your own antigens, but if I took your B cells and put them into my body, they would go crazy because there's all kinds of new antigens that they're not used to. So those B cells that are circulating in your body all the time are looking for antigens that will trigger that B cell receptor. Now, if you find an antigen and it binds to that B cell receptor, what happens? What happens to the B cell? So let's go back here to the T cells. That was kind of an overview of the B cells. Once you have B cells down, T cells are pretty easy, but because they do kind of the same thing. But where the confusion comes from are these MHC1 and MHC2. And it has to do with how T cells recognize antigen. So first off, where do T cells live? Where are they born? Born in the bone marrow. And then where do they mature? The thymus. But otherwise, the process is very similar. So T cells are born. They go through a process of genetic rearrangement to produce a T cell receptor. So the T cell receptors have as much diversity as B cell receptors. The biggest difference is that T cell receptors really only recognize proteins, whereas remember when I was talking about the antigens for B cells, it could be carbohydrates, sometimes lipids, or proteins. So B cells have more diversity in the antigens they can recognize. But T cells are very similar because they go through the same gene rearrangement and they end up with a very unique antigen binding site on that T cell. Then they go to the thymus, where they're presented with self-antigens, and any T cell that recognizes a self-antigen is induced to go through apoptosis. And so they die, and hopefully that means your T cells won't attack yourself. So that's the maturation happens in the thymus. If they pass that test, the T cells, just like the B cells, go into circulation. They go hide in the lymph nodes. They're looking for antigen. And they can be stimulated by antigen binding to the T cell receptor. However, that T cell, that antigen binding to the T cell receptor will only be recognized if the antigen is held by an MHC molecule. So we'll get to that in just a second. So that's one difference. B cells can be activated by free antigen. T cells, that antigen has to be presented to it by an MHC molecule. And we'll look at MHC in just a minute. So MHC plus antigen activates the T cell receptor. But just like B cells, T cells need a signal 2. So that is signal 1. Signal 2 for T cells can either come from an antigen presenting cell, which is what you're seeing over here. So here is your T cell receptor, and it's binding to an antigen that's being presented to it by MHC. So the purple is MHC. And we'll get to why CD4 is important in just a second. But then, so that transmits, once that binding happens, that transmits signal 1. But again, for a fail-safe, you're only going to be activating that T cell if there's a signal 2. So that signal 2 can come in the form of antigen-presenting cells or cytokines. And what do T cells do if they're activated? Is it because you don't know which type of T cell to pick? There's 2 types of T cells. So we have our B cell, and it's expressing this receptor. And all over. So this is a B cell, and we call this the B cell receptor. All of the B cell receptors on a single B cell are the same because they have the same genes, so they did the same rearrangement. So all 3 of these receptors that I've just drawn are identical, but they're different than B cell 2 over here. So he has a different receptor. So now these B cells have passed the test. They won't recognize self. They're circulating around. They go into a lymph node, and they see an antigen. And that antigen happens to bind to these B cell receptors. Is that enough to activate that B cell? No. Remember that antigen, which binds to the B cell receptor, transmits what we call signal 1. So that's the first signal. But that B cell is not going to be activated unless we have a signal 2. How will this B cell get signal 2? What was that? So cytokines from T helper cells. So T helper cells, there may be a T helper cell in the area. And cytokine activation is possible, but more directly is CD40 and CD40 ligand. You don't have to memorize the names of those, but there's basically a receptor on this T helper cell that binds to another receptor on this B cell. So T helper cell can directly bind, and that'll transmit in a signal. And these 2 signals together, the binding of the B cell receptor by antigen and the stimulation of the T cell to the B cell through CD40 ligand is going to transmit, both signals are needed for activation of that B cell. What's the second way we could give that second signal? What if there's not enough T cells around? Sometimes you really want those B cells to start making antibodies, but there's just not enough T cells around. What else could happen? Complement, yes. So remember that complement could also bind to a different receptor and transmit signal 2. So we have 2 signals needed to activate the B cells. That's going to be the same for the T cells. And when you look at this, it's a safety mechanism, right? So that first antigen means there's something foreign in the body, but everything foreign is not necessarily bad. So those of us with allergies know that hay fever or pollen or dust mites, they're not really bad for us, but they still can trigger a response because we have certain antigens that are recognizing it in the context of T cells. So we happen to have T cells and B cells that both recognize those antigens. But for the most part, we don't respond to foreign things that are not dangerous. So what signal 2 is it's supposed to say that you've encountered an antigen in the context of danger. So either that T cell has also recognized that something bad is in the body, or complement has been activated by the innate immune system. And the innate immune system is going to be activated by damage or microbes. So the innate immune system kind of gives a signal that there's a fight going on, so that the B cell knows, yes, I really should start to produce antibody. So we've had signal 1, we've had signal 2. Notice it's only this B cell that's gotten both signals. The second B cell over here, even if there's T cells around, it didn't bind to an antigen, so it's not going to be activated. So that's where the specificity comes for this system. 2 signals, now we're going to activate. And what does that mean? We go through a process called clonal expansion. So this B cell is going to start to divide and divide and divide and make lots and lots of copies of itself. And the reason we call it clonal expansion is because these B cells all express the same B cell receptor as that original guy. And this clonal expansion is partly stimulated by cytokines coming from these T helper cells. So the T helper cells do play an important role in activating B cells. They provide the co-stimulatory signal and they provide cytokine-induced proliferation. So now we have a whole clone of identical B cells that are going to start secreting free antibody. And we talked about the different classes of antibody that can be produced. So those antibodies are going to now go into the blood. They're going to circulate. If they're IgAs, they're going to go to the mucosal system. If they're IgMs, they're going to congregate. And those antibodies do a lot of things to help combat the specific microbe. But because it's only this antigen that activated it, and now all of these cells are going to recognize that antigen, all of these antibodies are also going to recognize that antigen. But no other antigens. Very specific. Does that make sense? Questions about B cells? One quick point. Some of these, during clonal expansion, most of these cells are going to become antibody-producing cells, but a few of them are going to become memory cells. So they differentiate, they've been activated, and then they're just going to hang out and wait until another antigen in the future comes by, in which case they're ready to go right away. So that's where this memory for the adaptive immune system comes in. So, okay. Right, so let's think about what these 2 different types of T cells do. Have you guys learned about helper T cells and cytotoxic T cells? Okay. What do helper T cells do?
They activate cytotoxic T cells and other T cells.
Right. They activate the cytotoxic T cells and B cells. So helper T cells activate other lymphocytes, the cytotoxic T cells, the B cells, and they do that by direct binding, like we just saw, so they have that co-stimulatory molecule that directly binds, But in addition, they can also secrete cytokines. And those cytokines can help activate cells. So they secrete a whole array of cytokines, interleukin-2, interleukin-4, interleukin-5. And the type of cytokines they produce will activate different types of cells. So they can help activate macrophages. So there's crosstalk between T helper cells and the innate immune system. And then they can help activate B cells, they can help activate T cells. So their main job really is communication within the immune system, both between the innate immune system and the adaptive immune system. What do the cytotoxic T cells do? Yes? Yes, they kill your cells. But not just any cell, of course. It has to be a cell that is infected with a microbe. So they're going to, if their T cell receptor is activated by a foreign molecule, then they will tell that cell, you have something foreign inside of you, time to die. So they kill infected cells. Yes?
I just have a question about the cytokines again. Does it secrete different cytokines depending on what it's infected with also?
It'll secrete different cytokines depending on what type of antigen presenting cell presents the antigen, as well as it gets really complex because there's a bunch of different subsets of T helper cells. So T helper 1 versus T helper 2 versus T helper 17 all secrete different cytokines. And those T helpers can differentiate from the beginning into these different types of T helper cells. And those T helper cells will only be activated in certain situations, and then they will secrete specific cytokines. So that's the long answer, but yes, is ultimately they secrete different cytokines depending on different stimuli in order to do different things and talk to different parts of the immune system. All right, T helper cells, what is the identifying molecule on there? CD4 or CD8? CD4. So this is really important when we're talking about human immunodeficiency virus. Because you'll remember HIV uses CD4 to recognize its host cell. And so HIV infects specifically CD4 positive lymphocytes. Whereas cytotoxic T cells express CD8. So these are markers that immunologists use to be able to differentiate these two cell types, because if you look at them under a microscope, B cells, T helper cells, and cytotoxic T cells all look identical. You cannot tell them apart. So you have to do special immunostains, and you can stain for CD4 and know that that's T helper cell. Or if you stain for CD8, you know that those are cytotoxic T cells. So we already said the T helper cells are stimulated by antigen plus MHC. The main difference between T helper 1 and T helper 2 is which class of MHC they recognize. So do you know T helper cells? Are they MHC1 or MHC2? Are they stimulated by any cell or by antigen-presenting cells specifically? So T helper cells are only stimulated by antigen-presenting cells which express MHC2. Whereas cytotoxic T cells are stimulated by any cell that's expressing MHC1. All of your cells in your body that will express MHC1 that has a foreign antigen. And then we already said T helper cells mainly work through cytokines, although they also have those direct ligands to bind to other lymphocytes. And then the cytotoxic T cells, if you remember back at the beginning of the last lecture I talked about, they induce apoptosis through chemical mediators called granzyme B and perforin. And basically, perforin pokes a little hole in the cell. So because you had that T cell receptor had to bind to the infected cell, those two cells are really close. And so as soon as it's triggered, it's going to secrete perforin, which is going to poke a hole in the infected cell and then deliver a bomb. Granzyme B is like a bomb that activates caspases to self-destruct, to trigger apoptosis. So let's talk a little bit about the difference between MHC1 and MHC2, because that's really important for distinguishing which cellular response you're going to have. What cells express MHC1? Just said. What cells express MHC1? So cytotoxic C cells recognize. Yes. So any cell of the body, except red blood cells, but just know every single cell in your body expresses MHC1. MHC1 is called major histocompatibility complex 1. Here's the protein structure for it. So you can see what it is it's this big, giant protein that has a little antigen right in the center. And so those T cell receptors on cytotoxic T cells, what they're looking for is this big site. It has to be antigen plus MHC1 in order to activate that T cell receptor. So this is going to bind to the T cell receptor on cytotoxic T lymphocytes. But that T cell receptor won't recognize that antigen by itself, and it won't recognize MHC1 by itself. It has to be the two together that will bind that binding site. So how do you get the antigen in there? Well, this is a really, really clever mechanism. What happens is inside the cell, all of your cells of the body, all the time, you're degrading proteins. You have normal protein turnover. So you're constantly chewing up your proteins through proteasomes. And all of those proteins, a little bit of them, little peptides, little pieces, are going to go into the endoplasmic reticulum, which is where membrane-bound proteins are made. Remember, you have that rough ER. So in the endoplasmic reticulum, MHC1 molecules will be made, and then all of these little protein fragments that have been chewed up are just kind of fit in. Plop, 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 plop. And then those proteins, those MHC1 plus antigens, are delivered to the cell surface. So what it ends up being is your cells are just displaying for the body a snapshot of what proteins are currently inside the cell. So all the time, all of your cells are just saying, here, this is what's inside of me. And it's a brilliant system because we've already eliminated any T cell that recognizes self-proteins. So the T cells that would recognize any of your normal proteins from a healthy cell are just going to say, I'm not really interested, and move on. And it's only if your cell happened to be infected by something, an intracellular bacteria or a virus, suddenly some of the proteins that get chewed up and put onto MHC are not supposed to be there. They're not self-proteins. And so when a T cell comes along, it says, hey, I recognize you. And if that T cell recognizes the antigen that's being presented on MHC1, you know that cell is infected. So clever, right? How else would you be able to tell the immune system these are cells circulating? They can't go inside all of your cells. So thinking back in evolution, that's how viruses evolved to hide from the immune system. They're like, well, I'll just go inside the cell. And then your immune system can't recognize it. So your immune system evolved a countermeasure to be able to constantly sample what's going on inside the cell. So that's what MHC1 is doing. MHC2 is very similar, but MHC2 is only expressed on antigen-presenting cells. So what antigen-presenting cells, this goes back to our dendritic cell. Those are the most powerful antigen-presenting cells. They're sampling extracellular antigens, right? They're constantly phagocytosing particles from their environment. And then they take those, they phagocytose them, they process them, they load them up onto MHC2, and then they put MHC2 on the surface of the cell. So antigen-presenting cells are specifically presenting antigens that have been phagocytosed from their local environment. And so that's how you can differentiate. That's how the body knows to differentiate, because you don't want the body, you don't want to activate cytotoxic T cells for bacteria that are extracellular, right? Because then you're just killing a lot of your cells, even though they're not in danger, they're not infected. If you have extracellular bacteria, you want to activate your T helper cells so that those T helper cells can help activate B cells to produce antibodies, so that they can help activate macrophages to elicit an inflammatory response. So that's why we have these two different classes of MHC. So MHC1 is to recognize and kill intracellular pathogens. MHC2 is to recognize and help kill extracellular pathogens. Does that make sense? Okay. So the main types of antigen-presenting cells in the body are the dendritic cells, the macrophages, and B cells. B cells can be antigen-presenting cells, although we're not going to get into that too much right now. So when you're studying this weekend, right, take a look at this, because this diagram shows you that process again, to refresh your memory about how antigen gets degraded and presented on MHC1 and MHC2. So the three antigen-presenting cells are the dendritic cells. Dendritic cells are professional phagocytosing cells that live in the epithelium and the mucous membranes in high numbers, constantly sampling what's trying to cross that barrier. Remember, that's your first line of defense for immunity, are those barriers. And these dendritic cells are there in case anything gets past. And so as soon as something comes past a barrier, you're going to start to trigger inflammation, the innate immune system. But at the same time, antigen-presenting cells are taking that information and presenting it to B cells and T cells to get the adaptive immune system going in case the innate immune system can't take care of it itself. And the second reason that you want to get the adaptive immune system involved right away is because, just a reminder, we've seen this before, activation of T helper cells is really important for the innate immune system and interacts with the innate immune system by secreting a bunch of different cytokines that activate macrophages. Macrophages can be antigen-presenting cells, so macrophages can also activate helper T cells. So you get this kind of cycle that happens there. Any questions about that? You know, it's a lot of information. Yes? The three types. So if you have a new infection, starting right here, right away, you're going to start to activate let's look at the gray line first here. So the gray line is the first time. So you're going to start to activate these cells. But remember, it takes 3 to 5 days for that process to happen. And that's because the antigen-presenting cell has to get to the right spot, that the right T cell, at any given period of time, you only have 1,000 cells that are going to recognize a specific antigen. So the right T cell has to find that antigen. So that circulating lymph is part of that system, but it takes a little bit of time. Once that antigen is presented and activates the cell, and you have the co-stimulatory signal, then the cells have to start dividing. You have that process of clonal expansion. And then once you've gone through that clonal expansion, now you have enough cells that can go out and do their effector function, whether it's B cells or T cells. So that's why it takes so long for the adaptive immune system to get going. But you're going to kill off that pathogen. Those effector cells are going to do their job. And then the number of cells is going to decline, but it doesn't go away. So those cells, once they've done their job, some of them are going to hang around, and they're going to hang out in your body all the time. So right now, you have T cells, memory T cells, that remember all the infections you had when you were a kid. That's why kids get sick all the time. But every time my kids get sick, I'm like, yes, because now you're building your memory. And they're not going to get sick as adults as often because they have good memory of all of these different pathogens. And so next time you have a new pathogen, those cells are sitting there ready. They've already know what to do, and they can mount a response right away. It doesn't take that 3 to 5 days anymore. So they can immediately start producing antibodies or secreting, going out and doing their cytotoxic function. So that's really the reason we have immunologic memory. And that's the theory behind vaccines, is that we want to stimulate this process without you having to get sick. So vaccines, we typically use them for infections that are very dangerous to people, like measles and mumps, rubella. We all have been vaccinated against that, because those are diseases that used to kill a lot of children. If you survived, you had great immunologic memory, but if you didn't survive, then that's, yeah. So if we can vaccinate all kids against it and try to induce immunologic memory without them having to get sick, then we can save a lot of lives. And that's exactly what's happened. Prior to vaccination and antibiotics, kids used to die. One in 3 kids wouldn't make it to their 20th birthday. And now that number is like 1 in 100. It's very rare that kids don't survive because of vaccination. Really cool. Okay, so that's a quick summary of what we've just talked about. We've talked about the different features for specific immunity. Remember, you should understand the difference between humoral and cellular and what mediators are in both. And then we went through B cells and T cells and how they recognize antigen and what they do. And I know it's a lot of information. Does anybody have any questions to put it all together? Okay. If you have questions, email them to me, and then I'll address them at the beginning of class, because I want to make sure that we're all getting the immune system. Before we move into transplant, let's take 2 minutes to stretch your legs. Okay. So the immunologic rejection becomes because you're recognizing the MHC of somebody else as different. They're expressing different MHC than your own cells are expressing. That seems to be the most immunologically reactive protein on your cells. So there can be 3 types of tissue rejection. And in general, tissue rejection includes both humoral and cell-mediated immune responses. So keep that in mind as we talk about these. So the first type is hyperacute. Hyperacute tissue rejection happens if you have preformed antibodies. So if in your blood you have antibodies that happen to bind to antigens on the donor tissue, as soon as that donor tissue is in your body, those antibodies are already there. They're going to swarm in. They're going to clog the blood vessels. They're going to bind to the tissue. They're going to trigger inflammation. You're going to have total rejection within seconds to minutes. And these preformed antibodies can come from if a woman has been pregnant, the baby is going to have different MHC molecules than her, because half of those came from the father. And so you will form some antibodies. I've had 4 babies, and so I have antibodies in me right now that recognize my husband's MHC molecules. And if I ever were to get a kidney from him, I would have a high chance of hyperacute rejection because I have those antibodies. The other ways you can get antibodies are from blood transfusion. So if you get blood transfusion, you can make antibodies to the transfused blood proteins. If you've had an organ before, so many times people who have a kidney transplant, they'll have chronic rejection, they'll need a new transplant. So they may have antibodies in their blood already that happen to recognize some of the antigens on the new donor kidney. So that's hyperacute rejection. However, we've gotten pretty smart about this clinically. And so whenever somebody is going to undergo organ transplantation, we take some of the donor cells and we take some of the recipient's blood and we mix them and we look for a reaction. So we can kind of tell if this is going to happen. 90% of hyperacute reactions are prevented by this cross-matching. So hyperacute, because it's preformed antibodies, is part of the humoral. It's a humoral response. Acute rejection can have both humoral and cellular. So with humoral, you don't have preformed antibodies, but very quickly after that transplanted organ gets into the body, you'll start to trigger B cells because they recognize these new tissue antigens as foreign, and those B cells are going to start to produce antibodies that bind and attack the donor tissue. Also, you'll start to stimulate T cells. So these new antigens that come in from the donor tissue will trigger T cells, and those T cells will come in and start killing all of the cells in the transplanted tissue through apoptosis. So you have both types. And you can have both types with chronic as well. Acute and chronic rejection, the mechanisms are very similar. It's really just the time frame over which it occurs. And we've discovered that the triggering of T cells, in order for this to happen, happens through both a direct pathway and an indirect pathway. So in the direct pathway, every tissue does have antigen-presenting cells. So when you transplant a kidney, some of that kidney, there's antigen-presenting cells in there. There's macrophages in there that are part of the donor system. And so those are presenting peptides, antigens, from the donor cells. So it's putting up all of these foreign peptides out there that your T cells come and say, that's foreign, that's foreign, that's foreign, that's foreign, and that elicits a big T cell response. The other thing that can happen is that when you transplant tissue, some of those cells are going to die, and some of the cellular contents from the donor tissue are going to be phagocytized by your own body's antigen-presenting cells. And so your own antigen-presenting cells can also present foreign tissue to the body and activate it. So those are the two types. That's what they mean by direct and indirect presentation. I'm going to leave it to you to go through these pathways, but this is just to reinforce how cellular and humoral immunity is activated in the context of tissue transplantation. But let's look at some pictures in the last few minutes of class. So here is a kidney that had to be removed because of acute rejection. And so here's the renal cortex, here's the calyx. And you can see this is actually really swollen and it's full of blood. So there's a lot of inflammation and necrosis going on in this tissue. That's what you see grossly. Now let's look at what you would see microscopically. So this is an image of acute cellular rejection in the kidney. So right here, these are normal tubular cells. And what you see over here are a lot of lymphocytes. You can see that they're lymphocytes because they have one big round nucleus that takes up the majority of the cell. The other thing you'll notice is some of these are plasma cells, where that nucleus is a little bit displaced and there's a lot of cytoplasm, dark pink cytoplasm around it. What are plasma cells again? What are they producing? Antibodies, right. So these are B cells that have already been activated and they're churning out antibodies. So you can imagine what's going on in this tissue. You have a lot of lymphocytes here. And you know that there's got to be a lot of antibodies in this tissue. So if you were wondering if a person has had a recent transplantation and their kidneys start to fail, you might do a kidney biopsy and take a look and see, oh, there's signs that there is rejection going on in this person. And in this case, we know it's acute because of the time course. Here's another example. Here's a glomerulus, so you can see the normal structure there. But all of these extra cells over here are inflammatory cells. So this is a sign of rejection. Chronic rejection usually includes a lot of inflammatory cells, but in addition, you're going to see scarring because this chronic inflammation. Remember, going back to what we've talked about, the difference with acute and chronic. So because you have chronic lymphocytes and macrophages coming into the tissue, and initiating the repair process, but then not going away because these antigens are constantly there and they're constantly foreign, you're going to get laying down of scar tissue over time. And the scar tissue ends up replacing damaged tissue. So most people with kidney transplant, over the course of 10 to 20 years, depending on the level of immunosuppression, are going to have scarred kidneys like this. And ultimately, usually they end up needing a second transplant over time. The other thing you'll notice in this picture is here's a blood vessel. And this blood vessel, the wall of the blood vessel is really thick. So that's scarring of the blood vessel. So another, with rejection, vasculitis is really common. And it's because your immune cells travel through the blood. And so when they're traveling through the new organ, they're traveling through the blood vessels, the first foreign cells they see are those endothelial cells of the blood vessels. So those are the ones that they're most likely to attack. So vasculitis is seen almost with every type of rejection. Here's another example of the heart tissue in a heart transplant, and you can see vasculitis. So these pink cells here are normal myocytes. You can see they have the long fibers, and there's their nucleus. So these are normal myocytes. But then you have all of these inflammatory cells. Here's a blood vessel that is engorged and has inflammatory cells around it. So this is vasculitis in then a heart transplant. What do you notice here? It's red, but not swollen. Yes, that's true. Does it look painful? Yeah. Yeah, so this is a rash associated with graft versus host disease. Graft versus host disease can be thought of as body rejection. Does anybody know what graft versus host disease is? Who gets it? What's that? So skin graft patients will get graft rejection, so the skin will be rejected itself, but graft versus host disease is actually widespread throughout the body. Yeah?
Bone marrow transplant?
Exactly. So this graft versus host disease happens with bone marrow transplant, because what you're doing with bone marrow transplant is you kill the immune system of your recipient, and then you put in a new immune system. So the donor immune system now recognizes the whole body as foreign. So people with bone marrow transplant have to be immunosuppressed. They have to take drugs that suppress their immune system, yet at the same time they need to have an immune system that works. It's a really difficult situation. So graft versus host disease is very common in bone marrow transplant patients. And what it is, it tends to happen in epithelial organs because they have a lot of antigen-presenting cells with a lot of MHC2 molecules in them. And so the skin, the liver, and the gut is where you'll see graft versus host disease a lot. The mechanisms, though, are the same as with organ transplant. The only thing is it's reversed now. It's that the donor immune cells are the ones recognizing the recipient's body as foreign. So here's a biopsy of skin in graft versus host disease. So here's the keratinized layer up here. And these are the basal cells in the skin. Down here is the dermis. And you can see that there's just a lot of inflammatory cells here. So this arrow right here is pointing to a cell undergoing cell death. What form of cell death do you think that is? Yes?
I'm just curious, how do you prevent that?
How do you prevent graft versus host disease? The best way is HLA typing. So if you can match your bone marrow donor to the MHC molecules of the recipient, you can really eliminate most of the disease. So the better the match, the better it is. And your ideal is somebody who has the exact same genotype at all 6 alleles for MHC1 and then also for MHC2. It's hard to find though, but there are databases out there. And periodically, in Boston and San Diego, I know that they'll do drives where you can submit your blood for HLA typing and you go into a bone marrow registry. And so that is a registry that's screened for anybody who needs a bone marrow transplant and who doesn't have a good match. And they try to find somebody who has a match for them. You're most likely to find a match in your siblings, because not your parents. Remember, your parents would only be a half match. Your siblings have a 25% chance of being a match because of the genetics. You're most likely to find somebody in your own ethnicity because of the shared genes. So whenever they do a bone marrow drive, they try to get a wide variety of people with different backgrounds in there so that you have the best possible chance of finding a match. So this cell right here is undergoing apoptosis, cell-mediated rejection. So the bone marrow of the donor produced T cells that recognized this cell as foreign and induced it to commit suicide. You can see that here too. This is the crypts in the gastrointestinal tract. All of these cells are being induced to commit suicide. And that, along with chronic inflammation, is what causes graft versus host disease. And just another example in the liver. All right, I think we are about done for today. So I will see everybody on Thursday, and we're going to do hypersensitivity and autoimmune diseases.